Thank you very much for inviting me. I think I want to connect to Craig's words before me, and I think I'll open my keynote by disagreeing with my relatively new boss, because that's a good way to start a day. I don't think there are 400 types of cancer. I think each cancer is individual and different, which is exactly why we all need you. Cancer really has become a data science. I realized its complexity and felt that I have the toolkits to try and help, and felt almost a moral commitment to do so, because cancer really needs a lot of data scientists. And as someone who's been here for about a year, one of the things that I can say that's very unique about this place is that the clinicians really do embrace the computational scientists and data scientists, seek them out and want to work closely with them in a way that I haven't seen in any other institution, and it's been great. Craig talked about the genomic revolution, the sequencing of our human genome, the mapping of all our genes and what it's done for science, and that's certainly been a major biomedical revolution that's really opened a lot of understanding in both health and disease. And we're in the process of another revolution. We've, why is this not working? All right, this is, okay. We've mapped out all our genes, but in order to understand what a mutation, what a gene does, how a diseased gene actually causes the disorder, typically genes are poorly expressed. Most of the mutations are actually in regulatory regions. That means that you have the wrong gene at the wrong time in the wrong place, the place being a cell. And while we have mapped our entire genome, we are very poorly mapped in terms of our cells and our cell types. We have an extreme positive information about what are the different cell types in our body, just characterizing them as well as what they do. We're in the 70s relative to where we were in sequencing land in terms of what we know about our cells. So recently, a bunch of us got together and decided that we were going to launch the Human Cell Atlas with the goal of comprehensively mapping each cell in our body, what it does, where it is spatially, how they transition between them, both in response to stimuli such as immune activation, as well as down a developmental trajectory. The consortium started in 2016. We have a white paper out that was recently published. And again, Memorial Sloan Kettering is going to be a big hub of this work, both in terms of the computational infrastructure for such a big project, as well as access to tissues at a cancer hospital. You can get access to even, nearby healthy tissues that you don't normally get. It's very hard to get a healthy or to get a kidney tissue out of a body or even more rare tissues. So towards the Human Cell Atlas, what's really been propelling it are single cell approaches. And in order to connect to this community, I thought I would tell about something that, this is RECOMB regulatory genomics. What it's really about is learning circuits and regulatory networks. And one of the things that single cells can do is that they can teach us about regulatory circuits. Really, one of the things you need to learn, one of the things that machine learning strives to have, is to have many data points. And single cell gives you exactly that. If you treat each cell as an individual, independent measure point, then current single cell data sets have now millions of single cells to learn from. And so if you treat each cell as an individual measure, and you want to know the relationship between the influence of gene or protein A on B, you can really use any trick that you want in learning functions to actually use the single cells as data points to learn the function of how the abundance of A influences the abundance of B. One of the first times this was demonstrated was actually with typical multicolor flow cytometry. This is 11-color flow cytometry, and in an experiment of one afternoon, and then through a Bayesian network learning algorithm, pretty much 2 decades of TCR activation was reconstructed automatically by a Bayesian network algorithm, as well as a new link at the time, which between ERK to AKT, which was subsequently validated. This is a stick figure picture, and as we try to go deeper and deeper and try and really actually add quantitation on these edges, not only to have a stick figure of who influences who, but how. What is the function? What is the computation that's going on here? We realize that single cells are very poor. That is because most of our learning happens as joint distributions. And most of the cells do a similar thing. And in this joint distribution, you pretty much have the peak completely dominating the signal right up in the center. So one of the switches that we did was switching from a joint distribution to a conditional distribution. What I'm doing here, people don't hear me? I thought I have a loud enough voice, but I will now make an even louder voice. Be prepared to sort of hold your ears and stuff. Oh, this is hard. It doesn't want to pop on. So I'm going to show you the exact same data, conditioning on X rather than joint distribution, and you can see what a difference this makes. What I just do. And suddenly you see the sigmoid. This is between CD3 zeta, and SLP76, two early stages in the TCR activation. And really what you do when you condition is really capture it on the full dynamic range. And so now you can look at the data before and after activation of the TCR, and you can see that after activation, the same increase in X, CD3 zeta in this case, leads to a much bigger increase in Y, its downstream SLP76, likely due to the activation of additional co-receptors that we haven't measured when calculating this relationship. Again, you wouldn't have gotten this if you would have looked at the joint distribution. You would have just seen a shift of the joint distribution from here to here. But here you can actually get the difference in the function of how X determines Y. So this is great, and this was done actually with CyTOF data. And in CyTOF you can get, again, millions of cells and pretty much every measurement that you want. But can we do the same thing for single-cell RNA-seq? Single-cell RNA-seq is wonderful. It's genome-wide, and rather than pre-selecting 40 proteins to look at, we can actually look at the entire genome and do genome-wide regulatory learning, and that would be great. The problem is, and the dirty secret of single-cell RNA-seq is that there's incredible dropout. So when you're looking at a profile of single-cell RNA-seq, you're not looking at a genome-wide full profile of that single cell. What you're looking at, depending on the technology, depending on the experiment, depending on the sequencing depth, you're looking at between 2 to 15% of the molecules in that cell randomly sampled. So this is actually a good data set collected by Ido Amit of mouse bone marrow. And you could look at some of your favorite immune subsets. For example, monocytes. They all should be expressing CD14 and CD11b. Yet only 2% of these cells you can detect CD14, and in only 6% of these cells you detect CD11b. This is because of that incredible dropout. This dropout particularly impacts gene-gene relations. So in the regular bulk genomics world, we have been used to doing correlations and more sophisticated modeling of dependencies between genes. In bulk sequence, that comes out, pops out, and has been the strongest impact of gene expression relationships. But when you sample a small percentage of the cells and you have a non-trivial chance of dropping out both X or Y independently, the probability of getting them both is pretty low. And this is a synthetically dropped out experiment. We dropped out 90% of the molecules modeled here, and boom, the relationship was gone. Most of it is on the X equals 0, Y equals 0, and the relationship is missed. One solution that some people have been thinking of is basically using CRISPR along with single RNA-seq, perturb-seq, crop-seq. There are a few others. The idea is that we're going to do some genetic perturbation and learn networks through that perturbation. Here, you add an additional degree of noise of CRISPR. You have to do an experiment that's pretty labor intensive. And of course, you can't apply this approach to patient primary data, which is my main interest. So we're going to have to do something computationally. And here comes magic. And people said I'd probably talk about magic because I really think that is a good introduction for computational scientists and how to view single-cell data. This is a work by Smita Krishnaswamy, who started this as a postdoc in my lab and is now an assistant professor at Yale, and David, a postdoc in my lab. Here's the same data as before. We're looking at 3 key immune regulators of development. Every dot is a single cell colored by its cluster, and we could figure out the cluster that it belongs to because we could look genome-wide and not just at these 3 genes. And this is the data before magic and without knowing anything about immune development. Oh, why is this not working? Ooh, this is the data after magic. Basically, magic uses information from nearby cells to reconstruct the missing values, reconstructing development, and you can see how one cell type after another, consecutive cell types along development from the hematopoietic stem cell all the way to the more mature erythrocytes. The reason that we can do this, the signal that magic is using, is actually the fact that our cell types reside on manifolds, reside on very regimented geometric objects. This is again Cytoff showing bone marrow with 13 colors with Cytoff, and you can see just how regimented this is. You have the same shapes. We've taken bone marrow from women, men, young people, old people. This is what these cell types look like. You can see that there's very clear geometric shapes, but of course, modeling this as a nice round convex Gaussian is again wrong. You need a non-parametric approach to do it. So the way to really look at single cell data is that it's not unconstrained. It actually resides on a very, very constrained lower dimensional object in this high dimensional geometry. And what we want to do is actually learn and utilize this lower dimensional geometry using tools for manifold learning. Here, Euclidean distance can be misleading because you're going from here to here through a region that there's no cell density, and you really want to stick and keep your computation within the cell density, within the lower dimensional manifold where the data resides. And in order to do this, what you can do is embed your high dimensional space inside a graph. This also removes the dimensionality. This also frees you from the curse of dimensionality, both in terms of computational burden and statistical burden of computing in high dimensional data. And basically, you take your high dimensional data and map it to a graph where every node is a single cell, and every cell is connected to its k most similar cells, the cells most similar to it in their genomic profiles. And now, instead of big Euclidean steps, distance between cells are measured as distances and walks along this graph. In order to get from one cell to another, you take walks or small steps within the manifold. If you're stepping to a nearby, very, very similar cell, you're likely remaining in the manifold. And through small, tiny steps, you can get from one cell to another. And this allows you, through different tools of diffusion maps and Markov processes, to really learn the structure of this graph. And now we can harness a lot of tools that mathematicians have been building for us in order to do this through a process called data diffusion or graph diffusion. And basically, we build this graph, which defines a distance metric and then an affinity matrix. And by raising it multiple powers, that's similar to diffusion on this graph. So let me show you this with real data. This is some data from the lab. Every cell is connected. The yellow dot is an edge on this graph. And as we diffuse, as we allow for walks, this is walks of length 1, as we allow for walks of length 2, 3, and so on, you can see a very, very distinct structure emerging. And this thing doesn't smear and just get all yellow, but it actually refines into a very distinct structure. Once you learn this structure, you could use the powered matrix times your noisy data to impute a cleaner data matrix, where every cell learns from its neighbors, weighted through diffusion directions along the manifold. So, I think it's misbehaving. So one thing I like to do is I like to check these things on synthetic data. So here's a developmental process from the worm. This is bulk data, which we're treating as single cell data, 206 developmental processes. This is the original data. We create false dropout and run magic, and we recover it. Temporal trend after temporal trend, magic recovers the synthetic data. And also in a systematic evaluation, magic is very well able to recover signals. After we kill most of the signal in the data, we can actually get very high degrees of correlation with the real data. Again, to show you on real data, this is again that bone marrow data, the diffusion happening. This is the raw data, so you could see in a sort of pairwise biaxial way, there are no relations. But as we diffuse the data and take more and more steps through the manifold, you can actually see the structure, the relationship between these gene pairs emerge and match the expected trends known from development. Here we looked at, again, a cell line. This is inducing the process of EMT, epithelial to mesenchymal transition, in a breast cancer cell line. The single cell data, as usual, it's a blob. With all the dropout, it's hard to discern shape and structure in the data. But once we run magic, there's very clear shape. Here we have our epithelial cells, our mesenchymal cells, actually some cells that are dying in between. And surprisingly, most of the cells are actually intermediate steps in the intermediary between the epithelial and mesenchymal step. And what we got is we get the entire transition from E to A and everything in between from a single time point due to the broad heterogeneity of this data. There are little details that matter. For example, that kernel that actually builds both the initial graph structure and the affinity matrix for the structure really matters. This is using standard approaches, textbook from the diffusion math theory. They really collapse all the data and don't get the structure. We use an adaptive kernel. The reason we need an adaptive kernel is that cell phenotypes have 5 orders of magnitude differences in their density. Stem cells are very, very rare, but some mature cell types, such as T cells, can be very, very frequent, and they would dominate unless we get this adaptive kernel that really adapts itself locally to the density and the frequency of the different cell types in the data. Again, standard kernel and what we get here with our adaptive kernel. Once we have the shape, we can actually, again, we have a geometry, we have a manifold, we have reconstructed the shape. We can now study the shape. And one of the surprising things we found is that it's actually not one simple trajectory through a straight line from E to M, but there's actually a very, very high-dimensional complex intermediate state that actually resides in, based on our approximation, about 10 dimensions. And we want to study this complex shape of the intermediary between the E to M, and we could use archetype analysis, finding the edges, the extreme points of the shape from beyond which biology doesn't allow you to go. We could identify these 10 archetypes. Right here is the mesenchymal archetypes. These are 2 separate epithelial archetypes and a bunch of intermediate archetypes. And now that we've defined these archetypes, again, identifying the edges of the data, the extreme edges of our geometric object, we can study what are these extremes and what differs between them. And we particularly wanted to look at some of the extreme intermediary states. And we could look at differential expression analysis in each of these archetypes, and we could see a very large number of genes had very different distributions of gene expression in a whole slew of genes. There were about 6,000 genes that were significantly different, as you could see from this rough sampling here. We could define what really characterizes each archetype, and we saw that each archetype had its own set of transcription factors that really defined it, its own set of chromatin modifiers. We could see right here a bunch of archetypes that had many features of stem cells that were also very active in heavy chromatin remodeling, suggesting that the cells revert back to a stem-like state. and then go forward to the new mesenchymal state. But really what this allows us, again, not only to capture the structure of the data, but look inside at the gene-gene relationships. This is, for example, EZH2, a key factor in chromatin, and you could see before magic there's almost no signal. It looks like noise, and after magic you see that this goes up in these intermediary states. We found a very large slew of genes, including both transcription factors and chromatin factors and signaling molecules that go up exquisitely in the intermediate state. They're low in the epithelial state, they're low in the mesenchymal state, but they're very, very active in between. And here's a bunch of other relationships, again, garbage, including ZEB1 and SNAIL1, the known transcription factors of this transition, which behave as expected and have structure after we run magic. Of course, staring at objects is not fun, because the number of possible gene pairs is too high to stare at them. Therefore, you want to actually quantify this. And we use this idea of conditional distribution to actually build a quantification. We figured out the density of each and every one of these dots. And we really have combining magic as well as the conditional. This is before the conditional and this is after the conditional, to really find and quantify and visualize and vectorize these gene-gene relations. This allows us to now cluster them, and now we can actually get a full map from early genes to late genes in the transition. This is cells sorted by Vimentum, which is a key marker for the EMT, and the waves of factors and genes that rise and fall one after another through the process of EMT. And this is basically a global map of this dynamics and what genes rise and fall, including regulators, as this dynamics is occurring throughout the process. By having this dynamic map, now we can actually try and learn gene-gene relationships. We could use this pseudo-time, because correlation doesn't give you causality, but temporal time gives you an causality. And even though we have only one time point, because we ordered these in a sort of inferred pseudo-time, we're going to use that as time. And we're going to use our DREAMY score that looks at the quality and the mutual information of the conditional distribution. And together, we're going to use this to infer a list of targets for each transcription factor that's active in the data. So let's start with ZEB1, which was of particular interest to our collaborators because ZEB1 actually has a very, very poor antibody for CHIP, so its targets weren't well known. We had our list of predictors. We actually now overexpressed and activated EMT through a ZEB DOX-induced overexpression. And if we use timing alone, we see that we don't have a significant overlap between our predictive targets, meaning while ZEB1 is a key transcription factor for EMT, it's not controlling all the changes that we're seeing. The mutual information between ZEB and its targets gave us something significant, but not quite, because there are many other factors. Again, correlation, information, statistical dependency does not equal causation. But when we looked at the temporal process, when we used both time and mutual information together, we got a nice p-value that I like in terms of the hypergeometric overlap between our predicted targets and the targets that raised up following the ZEB induction. This is one. I wanted to do all of them. I didn't want to build cell lines for 300 different factors. We're largely a computational lab. So here we switched to ATAC-seq. And we used ATAC-seq as another manner in which we can make predictions. Again, it's not perfect, it's predictions. And particularly ATAC-seq doesn't actually look only at activators. It looked at poised genes, it looks at repressors. And ATAC-seq isn't the perfect proxy. But now I have two independent predictions of targets using two independent sources of data and technologies, and I can look at their overlap. And the overlap was larger than expected for 291 out of 292 of the transcription factors that are active in the data for which we had predicted targets. And this is significant for 268 of these targets. So we actually built, from unperturbed data, can do a global network involving about 300 transcription factors and 10,000 genes at high degree of accuracy just by treating the single cells, each on their own as data points, and combining that with slightly more sophisticated computation. So in conclusion, MAGIC is a very powerful tool. I recommend you use it to really fill in the missing gaps and make your data more interpretable. It's been available for a while now. People keep on emailing me how it's really revealed the biology in their data. People are very happy with their MAGIC data. The biology that's inferred through the MAGIC data in many different systems now has brought out and been validated. And you can take this idea all the way to reconstructing regulatory networks from single-cell data, from single samples and single snapshots of regulatory data. So let's take a little bit of this idea, a little bit of the infrastructure that we have for looking at single-cell data, and see if we can do something real. We're at Memorial Sloan Kettering, so this was a wonderful collaboration with Joan Massaguet, who's truly an expert in metastasis and a great colleague, and Ashley, who's become a joint postdoc between the two of us, really the next generation that takes all the experimental and biological understanding of metastasis, all the computational training that my lab has to offer to really make an impact on understanding something such as metastasis. So while most of the computational biologists are looking at TCGA and some of the CCLA lines, and many of the big data sets really focus on bulk, really focus on most of the cells, actually what kills is metastasis. This nasty little tiny fraction of population that remains, even if the therapy really kills most of the tumor, 99.99%, you still have that nasty killer cell remaining. The cell can be latent. All of us have been impacted by cancer where we think we're home free, and 6 months, 2 years, sometimes 5 years later, the worst occurs and the cancer occurs. And this is really the fact that a lot of cancer remains, and the nastiest type that remains are these latent cells, these silent cells that hide inside the body and lie sleeping undetected for months and years until they recur far more aggressive than the first time around. These have some stem-like properties because they can grow into a full tumor. They're able to evade the immune system that's designed to kill them over months and years, and they have competence in invading multiple organs. And it's really trying to understand this latent stage that's what we need, and a focus of Joan Massaguet's lab, who's the world's biggest expert in this process. And while postdocs are usually impatient, and they like to judge metastatic models as quick as possible, you want to do your experiment, you want the metastasis to break out as quickly as possible, you want to inject the cells, and a week later, 2 weeks later, be able to do your experiment. Actually, if you want to model metastasis in biology, if you want to recapitulate how it happens in patients, where it recurs after months and years, you actually need to develop a model where this is the case in mice, where you have cells that after you inject them, they are indetectable for at least many months. And this is a model that Joan developed in his lab, published here about 2 years ago in Cell, and has been a model that many people in his lab, including Ashley, have been studying. Again, you could use the mouse itself as a flow cytometer. These are metastatic cells that you can inject in the mouse, and then you can't detect them, they disappear. But when you sort them, you could see that they have these little fluorescent labels. And one of the things I want you to notice, because they're colored, and they're colored with rainbow, you can actually see that most of the metastases that do break out are clonal. They come from a single clone, a single color, and this is going to be interesting in the rest. These actually can take months, many months. Often they don't break out before the mouse dies. Mouse are short-lived, but there are sometimes mice that break out spontaneously after 6 to 9 months, and we looked at a few of those, even though these you have to grow the mouse for a while. And if you're impatient, if you remove the natural killer cells, they immediately pop out. It's the natural killer cells that are holding these at bay, and by removing the natural killer cells, boom, the metastasis pop out. And we could look at both these scenarios with and without the natural killer population. So looking first at the natural killer depleted cells, we can ask what is the heterogeneity of the populations that emerge. And remember, these are clonal. And what we see is incredible heterogeneity. We have 20,000 cells here from 8 different metastases across 4 different mice. And you can see with the T-SNE map an incredible heterogeneity that you can map down to a number of clusters. This is clustering with phenograph, and you see this incredible diversity. One way to try and interpret this diversity is really to use curated signals from bulk data, as well as curated signals even from single cell data clustered from the healthy. I really believe that in order to understand the disease, you first have to get a good map of healthy, which is why I'm so involved in the Human Cell Atlas, which is first mapping a healthy map of our body. Just like in a TCGA, we first mapped the human genome before we saw what mutations occur and what's different in cancer. And one of the surprising things that we saw is actually many of our cell types actually recapitulated many known cell types, or cells that were very, very similar to most of the lung lineages, both the proximal and distal lineages. So from a single clone, we could actually get something that recapitulated and mimics lung development of many different stages, of many different parts of the lineage. But this varied quite significantly between each of the MET. So each of the MET had cells from almost every major lung cell types in quite different proportions in the lung. And remember, these are clonal, and this full mimicry of lung development came out of a single, very plastic clone. We wanted to compare that to the spontaneous outbreaks. These are outbreaks that managed to escape the oversight by the natural killer cells, managed to break out into a full-blown metastasis with the natural killer cells who are meant to guard the body against this intact. And what we had is we had a few cases where we could actually sort out these cells before they outbroke. We can get a few spontaneous metastases that outbroke by themselves without depleting natural killer after sometimes 6, 9, even in one case 11 months. And in one case, this was actually supposed to be a control. This was actually supposed to be metastasis-free, but we were lucky and found a little micromet that had already had a substantial size that was undetectable. And so we could have this small intermediate stage. And what we saw is that the development recapitulated from the proximal to distal. Proximal is right up here at the top of your trachea. Distal is the cell types closest to where the oxygen exchange happens. So you could see that this process, as these as these latent cells outbreak, match a transition from the proximal to the distal, with this intermediate state being here, this very, very stem-like state. SOX2 is the major regulator of all the proximal cell types. SOX9, which completely dominated these spontaneous outbreaks, was the primary regulator of the distal. And unlike the NK depleted cases, where we saw both proximal and distal cell types all mixed together, in the naturally occurring outbreaks, we only saw distal phenotypes represented. So you have a mimicry, again, of development through the process of metastasis from proximal to distal, as well as a metastatic progression right here. And really, what we actually quantified this and said, is there more diversity in the NK depleted versus the non-NK depleted? And we quantified this, again, keeping and accounting for the size and doing a lot of tests to really account for it. And you see that the NK really limit and keep at bay many phenotypes. So without the NK cells, the heterogeneity and the type of phenotypes that these single cells could occur in the metastasis were much broader and diverse. And the NK cells really tailored and pruned and limited the phenotypes that could survive as these spontaneous METs occurred. Now that we know what we're looking for, now that we studied this in a very controlled mouse where we can know what we're doing, we can perturb it, we now had enough information to try and see, well, what happens in human? Fortunately, I don't have a tail, I don't have whiskers, and I live more than 3 years. Humans and mouse are not the same thing. And one of the wonderful things about this institution is once we had a good idea of what we wanted to do with primary cells, actually went across the street to the hospital, talked with Charlie Rudin, who leads the lung service, and said, I want samples from tumor resections. Can you give me some? And within a few weeks, Ashley had 8 primary lungs, 4 metastases from different locations, as well as 4 normal samples from adjacent that were pathologically normal. So we can, within a few weeks, through our collaboration with our very engaged clinical friends across the street, really look now into real patient material. This is fresh stuff, cut out of a patient, rushed across the street so we could process it as quickly as possible. And with single-cell data and with primary samples, you want to rush. And before I moved here and I was at Columbia, we could really see the impact of that 25 to 40-minute cab ride on the quality of our data, which has now really improved thanks to the fact that it's now a 2-minute sprint across the street. And now we could look at it. The primary samples we asked for very early. So these are very early stage lung adenocarcinomas. These are stage 1 and stage 2. Again, a wonderful thing about Memorial Sloan Kettering is because of the volume, we could ask for pretty much any type of crazy sample we want that's typically rare at other places and frequently here due to the volume. And one of the things you see when I map this, and this is very important, is that the patients are well mixed. When your data is bad, when your experiment is bad, you see huge batch effects, you see huge blobs of one color. Each cell here is colored by its patient ID. You can see massive batch effects. And when I see this and I see everything is all mixed up and the samples are well mixed up, I'm happy. I feel, okay, this experiment succeeded. The first thing we did is we clustered. Again, we found a large number of clusters, a huge number of diversity. Most of the diversity was actually myeloid cells, T cells, with actually a very small number of epithelial cells, largely lung cells, and a little bit of stroma. And at first we thought we failed because we had so little tumor cells, and it was all immune cells, and we thought, well, maybe our epithelial cells were dying out. So again, we went across the street. They measured the frequencies before our entire process. And again, based on facts and other measures, these frequencies were, as well as TCGA clonality, these proportions were actually right. Lung tumors are hugely rich in immune cells. So now we could see which of these things match. And we can ask, do we see the same type of mimicry that we saw in the mouse in the lungs? And indeed, we focused on these epithelial cells because we wanted to focus on them. We used data from now human samples. We used our normal. Remember, we have 4 normal samples. So we used the 4 normal samples to build a good map of normal cell types. And then we compared these clusters from the normal sample that has very, very distinct phenotypes here and matched them to our tumor samples. And we saw, again, each and every one of these tumors, both the primary and the MET, really recapitulated many of the normal samples, particularly the primary had all of them. And really we see a strong developmental mimicry also in real patients. What about this metastatic outbreak thing? The fact that natural killer cells really trim and prune the possible phenotypes, the SOX2 and SOX9 dichotomy between the proximal and distal. Again, we had 8 early stage primaries and we had 4 METs. And we could see that in the primary we had many cells that were both SOX2 and SOX9, and we had the full gamut of natural lung phenotypes in our primary tumors. But in our metastasis, SOX2 was pretty much 0 or close to 0 for every cell, and SOX9 was really the dominant transcription factor in each of our lung samples. And finally, lung is a very heavily metastatic disease. Most lung patients don't really fare well. And we asked, what happens? do these latent metastatic stem cells persist and pre-exist in the tumor? Are they there from the beginning when they're still stage 1 and stage 2? Or is it something that's induced after dissemination? Is it an evolutionary process that happens later on? So because from the mice we can get a very, very, very good signature for what these stem cells look like, because we labeled them fluorescently and could measure them before they outbroke, we could actually get a very strong gene signature for what these metastatic stem cells look like. And we could look inside our primary lungs. These are, again, early stage lungs. And we could see an incredible enrichment of the signal for all these metastatic abilities already in stage 1 adenocarcinomas, bearing some sad news. But if we could figure out, now that we've isolated and characterized them, if we could figure this out, then we could target them directly while we are targeting the main tumor. And again, what we could do here, we could look at the primary tumor in patient samples, we could look at the metastatic outbreaks in patient tumors, and we used a mouse model to really understand this latent phase that's less accessible in patients, because it's really hard we can't really label cells and have humans walk around with engineered labeled fluorescent cells in their body. So again, showing you a demonstration of how we now took some of the techniques I showed you theoretically in a cell line and really apply them to understand these cell types in a clinical setting. Now, I knew I wasn't going to get to this, but I always like adding slides because I'm always optimistic in how much I'll get through. And just really leave it at acknowledgments. A lot of development was with David and Smita, who really developed magic. Ashley has been a wonderful conduit for a great collaboration with Joanne Massague and the people at, across the street, as well as a huge team of people in my lab who've been really developing the framework to really look at single cells in a very accurate way. And there's position available here in my lab and all across the institution. We need computational scientists everywhere on the clinical side, in my program, in my lab, and in this institution. Thank you.